All right. So if we talk about a hydrogen ion pump, how is it going to move hydrogen ions? All right, two, right? It's going to move against their concentration gradient. It's going to be from lower to higher concentration. Because the key word there is it said pump, right? So you know it must be an active transporter. And the active transporter is always moving things against their gradient. Make sense? The clue was in the question. All right. Okay. So we have other ways to describe how pumps work that have to do with how many solutes they transport. Okay, so we've talked about classification based on energy use, right? Active or passive, pump or transporter, right? We can also talk about how many solutes are transported in which directions. So if a transporter moves something only in one direction, we say it transports one thing, or we call it a uniporter. Right? Sounds like what? If it transports two things, all right, we call it coupled. All right, wasn't our sodium potassium a coupled transporter? Because it was transporting sodium and potassium. If it transports both things in the same direction, sim means same, we'll call it similar. If it transports things in opposite directions, anti means opposite, we'll call it an antiporter. So was our sodium potassium pump a sim porter or an antiporter? Anti, right? They were going in opposite directions. All right, so we have lots of different ways to describe transporters. This is another way you might hear a transporter described. By how many solutes and which directions they're transported. So remember that we said there was different ways of using energy to drive transport, and our sodium potassium pump directly used ATP. Okay, and directly using ATP. Using that ATP, it keeps the concentration of sodium outside the cell really, really high, right? That's like water behind a dam. It's potential energy. Right? The cell can now use that sodium gradient to drive other coupled pumps that are in the membrane. So instead of using ATP directly, okay, it can use that sodium gradient. Just let a little bit of water out from behind the dam to drive the transporter. Okay, so here's our sodium gradient. All right. Here's a glucose gradient. It's going the opposite way. All right, in this situation, we want to be able to move glucose into the cell because the cell needs to use glucose for energy. All right, so we're trying to keep the concentration of glucose in the cell high. We're accumulating glucose. All right, so what's going to happen here, here's our transporter. Okay. Our transporter is going to bind both a glucose and a sodium. And by letting the sodium flow down its concentration gradient, it's using the energy of this gradient to drive the active transport of this glucose against its gradient. And so it's going to bring both of these into the cell. And then the glucose, right, is going to get used for energy, and our sodium is going to get pumped back out by the sodium-potassium pump. So rather than using ATP to drive this, the cell is using the gradient that was set up by the sodium-potassium pump to drive this. So ATP had to be used at some point, right, to set this up. But we don't have to use ATP directly to transport this glucose. So sometimes you'll hear this called secondary active transport. Sorry. Right, and that means that ATP was used at some point to set up a gradient, but the ATP is not being used directly for this transport. Right, questions about this? Yeah.
So is this passive or is it active?
It's active. That's why it's called secondary active transport, because the energy is coming from this concentration gradient. That's the energy. So it is using energy. Other clarifications? So you might be asking yourself, why do we care? Why is this important? Think about the breakfast that you hopefully had before you came to class, which is now in your small intestine. So here's the inside of your small intestine. The job of the inside of your small intestine is to absorb the nutrients from your food. Right? These would be things like glucose, which your cells are going to use for energy. Right? This is a place where your sodium glucose transporters are going to be. They're going to be performing the secondary active transport to bring glucose into the cell. All right. This cell is also, of course, going to have sodium-potassium pumps to bring that sodium back out of the cell. All right. You're also getting, right, some of that sodium from your food, because your body needs sodium. All right. So you're bringing the sodium into your cells, and your sodium-potassium pump is putting that sodium into your extracellular fluid. All right. And this cell, right, can then also release that glucose, so it can be transported, like, to your liver to be stored. All right, so transport, really important in your small intestine. Yeah?
Isn't the sodium potassium pump also pretty important with cardiac function as well? Or
So, ion concentration is always going to affect muscle contraction, and that's going to affect the heart. We will eventually talk about muscle contraction. All right. So, other things that we've recently talked about that are really important to this kind of cell. Here are tight junctions. Remember, the tight junctions are a diffusion barrier. Can this transporter diffuse to the other side of the cell? Why wouldn't you want the transporter to be able to move to the other side of the cell? Why is it important that the transporter stay over here? Yeah.
so we stuff from that side, I guess like that will to come in.
Right. Is the food that you eat going to be over here? No. How are you going to bring in nutrients if this transporter is over here on the side of the cell? You're not. It's extremely important that your transporters for bringing in glucose and sodium are on this side of the cell and don't diffuse over to the inside of the other side of your cells where they're exposed to the extracellular fluid of your body. Here they have to be in contact with the inside of your gut so that the nutrients from your food can actually be brought into the cells. Right? How else are nutrients going to get into your body? Right? They're not. You're going to starve to death even though you're eating food if your transporters aren't on the right sides of your cells. All right? So whether or not these proteins can diffuse in the membrane is very important, right, to the function of this cell type and the rest of the cells of your body. Right? Questions about this? Yeah?
Earlier you were saying how something only happens in algae and bacteria. Were you saying like they're the only ones that use light as their energy?
They're the ones that use light, like so plants,
Yeah.
right? Algae, bacteria.
I wrote the wrong
Yeah,
thing.
that's okay. Other questions? Okay. All right. So, right, we're always talking about how structure is related to function. We talk about that in terms of the structure of a protein. It's also true with the structure of your cells, right? These intestinal microvilli that are here on the side that's facing your gut, these are here because they increase surface area, which means that you can put more proteins here that can absorb things and absorb nutrients, right, from the food that you eat. So they increase the surface area and give you more space for transporters. Structure and function are related. Okay. So we've just been talking about how in animal cells, the sodium-potassium pump pumps all the sodium outside the cell, and then that sodium is used for other transporters to do secondary active transport to allow other things to be driven into the cell. Okay? Other types of cells use that kind of system as well. However, most of them do not use sodium ions. Okay, so for example, plant cells use hydrogen ions. So they will pump hydrogen ions out of the cell, and then use hydrogen ions to drive their secondary active transport. Okay, so, and other organisms are going to do things differently, and maybe use different ions, but the basic principle is the same. We create a concentration gradient of a particular ion, and that's part of the physiology. Then that concentration gradient is used to drive transport of other things. Okay, and then this table here is just for your information, examples of other pumps. Okay, don't memorize it. Okay? All right. We talked a lot about transporters. Let's talk about channels here for a second. All right, this is a potassium channel. A lot of people kind of think of a channel as like a hole in the membrane that just lets things through. That is not true. Okay, it is a protein. Right? It has structure. It also has specificity. So the channel, right, only has an opening that's of a certain size and a certain charge. And it's only going to be able to let certain ions through. So for example, potassium here is of the right size and the right charge to fit through this region of this channel that's called a selectivity filter. That's the thing that's going to make sure that other ions can't get through this channel. Okay, so the channel is not just an open space in a membrane. It is very selective for the certain ion that is allowed to pass through it. The other thing is that these channels are not always open. All right? They have these things that we call gates, and they can be closed. All right, so this is closed, this is open. All right, and there are different things that can control whether the channel is open or closed. All right, different things. All right, some of these channels might be controlled by charge, that is voltage. And they may only be open under certain voltage conditions. Other channels may only be open when something binds to them that comes from outside the cell. So that's an extracellular ligand. Others may only open when something from inside the cell binds to them. And others may only open under certain mechanical conditions. So that's a mechanical stimulation, like a vibration or a movement. So channels, very selective, not always open, only open under certain conditions. Questions? All right. Okay. So let's talk about a specific type of transporter as an example. So we did some general principle stuff. Let's look at a specific one. So the specific one we're going to look at is called a family that's called the ABC transporters. And they're called ABC transporters because they have a region in them that's called an ATP binding cassette. So guess what? They bind ATP. And these transporters are found both in eukaryotes and prokaryotes. And what they do, of course, depends on the specific transporter. This is a whole family of different proteins, and you have to be looking at a specific one, right, to know exactly what it does. All right. Here's the general structure of an ABC transporter. All right, they have two transmembrane domains, and they have two of what are called the ABC domains that you can see bind ATP. Sometimes they're called nucleotide binding domains, or NBD. All right. So here, in this particular transporter, this is outside the cell in this picture. The thing that it's transporting is right here. As it's coming through, you can see that our transporter is bound to ATP. The ATP is hydrolyzed, conformation changes, our substrate comes through the channel. Okay? All right, so we've looked, this is our general principle, right? We talk about transporters or channels. Which one of these are you going to call this? A or B? Transporter or channel?
this side was hydrolyzed because it was broken.
The ATP is hydrolyzed,
And
right?
then it's broken apart, right?
Yeah, so one of the phosphates comes off the ATP and it's converted to ADP, right? That's the energy source.
Yes.
So active or passive?
Um,
Active, right? Used ATP. Okay. Okay. All right. Defective transporters can cause serious disease. All right. So you may have heard of the hereditary disease cystic fibrosis. Individuals who have this disease are homozygous, recessive, for a defective cystic fibrosis transmembrane conductance regulator gene. And for short, because that's a mouthful, we call it the CFTR gene. This gene encodes an ABC transporter. So people who have cystic fibrosis have two alleles that encode defective copies of this transporter. And this transporter transports chloride ions. This is a very serious hereditary disease that affects the entire body, but mostly the lungs. Causes a buildup of mucus. People get really sick. They get a lot of lung infections because of that mucus. And most people don't live past the age of 25. Now, everybody, right? Everybody has the CFTR gene. Everybody transports chloride ions. But if you don't have cystic fibrosis, that means you have functioning wild-type copies of the CFTR gene. At least one, right? You're either heterozygous for a functioning copy, or you're homozygous for functioning copies. Right, so everybody has the gene. But people who are affected with cystic fibrosis have two recessive copies of a mutation. Does that make sense? Going back to genetics here for a minute. You good so far? All right, so we talked about the ABC family. Now we're talking about the specific example in the family. All right, so this up here, right, if I was drawing a 2D diagram of the CFTR gene, this is what I would draw. Okay, so here's the one of the transmembrane regions and one of the nucleotide binding domains. Here's the other transmembrane region and the other nucleotide binding domain. This region in red here, this is where it gets phosphorylated. So not only does it have to hydrolyze ATP, it turns out that the CFTR protein also has to get phosphorylated for it to work. So it has to use ATP, and it also has to get phosphorylated. Okay? And so then here's the ribbon diagram of this structure. So everything that's in blue here is in blue here, and everything that's in green is in green, and then this is the R domain here. All right? All right, we're not trying to like draw anything or memorize anything here. It's an example. Okay, so we were saying that to transport these chloride ions with our CFTR protein, the protein has to be phosphorylated, and it also has to hydrolyze ATP. A little bit easier to see in this diagram. So here's our two transmembrane regions. Here's our nucleotide binding regions. All right, here's the chloride ion that's going to be transported in green. All right, here's the regulatory domain in purple. All right, notice this yellow, this is where it gets phosphorylated. It has to be phosphorylated for it to be able to open. All right, you see how it's bound to ATP here? There's another spot for ATP to bind. That other ATP binds and gets hydrolyzed. And this is what allows the chloride ion to be transported. This channel then opens, the chloride ion can be transported. So you need both the phosphorylation, binding of ATP, and hydrolysis of the ATP to allow the transport of the chloride to get out of the cell. All right, so specific example of a transporter and how it functions, right? Now, what does this have to do with the cell and how the cell is functioning? All right, so here, we're looking, imagine inside the lung, and we're thinking about the epithelium of the lung. And remember, when you breathe in, right, that air is like going into hollow sacs, right? So this is the inside of those hollow sacs, right? And these are the cells that are lining those sacs. And this surface is touching the air, but of course, cells have a little bit of fluid on top of them. All right, our conductance regulator, our CFTR protein, is on this surface. And it's supposed to be moving chloride ions out when it's functioning properly. That's what it's supposed to be doing. All right. If you have defective CFTRs, then these chloride ions cannot be pumped out. And what happens is you have less water